bring you right into the forest and look at the closer look at the trees. So specifically, I'm going to take a look at the the details in the lung organ, the lung oncology. So the objectives include determining the appropriate lung cancer patient for NGS sequencing or other molecular testing, and also interpreting the results coming from that. So expected after this talk, hope you know why we're doing this test, whom we're going to test, and test what, and when, and how. And in the last few slides, we're going to talk about the future challenges and potential solutions. As we alluded to, precision medicine is not a one-size-fits-all. It's just the opposite. So it's always about deliver the right treatment to the right patient at the right dose and at the right time. Because of the realization that the somatic mutation is the foundation of cancer development, therefore, we realized the marker screening through genomic analysis is the key for this process. However, we also realize testing is very expensive, and not everybody will benefit from testing, at least for now. Therefore, we were always interested in looking at other demographic evidences or tumor characteristics which can suggest this testing might be beneficial. That is why we're doing this. So starting from why we're testing, I think because the lung oncology is exemplary in the advancement of precision medicine. And I'll bring you to this very first clinical trial conducted by Dr. Mok in Hong Kong Chinese University, which provided the strongest evidence that we should be testing. This trial started in East Asian population that has enriched EGFR mutation. What this study did was taking that population and randomize them to receiving either chemotherapy or EGFR inhibitor. And what they find is that initially they did fairly about the same. With time going on, actually the EGFR mutated folks did better. Further analysis showed that of those patients who carried the EGFR mutation, if they received gefitinib, and they did much better than those who were put on chemotherapy agent. And of course, for those patients who did not have a EGFR mutation, if they're given the gefitinib, they did much worse. And these kind of patients will benefit to first-line chemotherapy. So this is perhaps the strongest evidence to show that this testing for EGFR mutation at the first-line setting is strongly recommended. The latest evidence that support this kind of practice coming from the FLORA trial. In this case, the third generation EGFR inhibitor is given along with the first generation inhibitors. And in the end, you can see from the survival curve, the third generation performed much better than the first generation. The reason behind that is the recent finding that the current molecular testing has its limitations. Oftentimes, what we find is the dominant clone, and we don't know there is some minor clones that exist in the same tumor, but we have not detected. However, with the treatment, especially with the first generation EGFR inhibitors, those clones will thrive and eventually become drug resistant. Therefore, they compare with the third generation inhibitor, they have a poor outcome. So these slides shows you the original AURA3 trial, which is showing that the osimertinib is effective in EGFR mutant that acquired resistance after the initial EGFR inhibitor treatment. Since the discovery that ALK translocation is driving a portion of lung cancer growth, we quickly developed a drug that targeting this lung cancer. As you know, that is the first and the quickest we have ever brought to the clinical arena, the crizotinib. Since then, we have made a tremendous advance. We get approved that ceritinib, when the patient become resistant to crizotinib, the latest evidence is showing a marvelous response is alectinib, which is also ALK inhibitor from Japan. In this study, the ALEX study, the patient who received alectinib instead of first generation crizotinib, which is a dual inhibitor of MET as well as ALK, had a much better outcome than the first generation. And showing here is those patients who had incidence of brain metastasis. You can show here those who received the alectinib had much lower evidence of developing brain mets compared to those who received the crizotinib. So small molecule inhibitor is not alone. So when we talk about precision medicine, immune therapy, which is also our targeted therapy, and is also bring this to a next level. So in the famous Keynote 24 trial, which compared pembrolizumab with chemotherapy as a first-line agent in those patients who express PD-L1 at 50% or above level, astounding differences we're saying here. So those patients who received the pembrolizumab had a much better chance of disease-free survival compared to those who received the first-line chemotherapy. So that essentially established the rule of pembrolizumab in those patients whose tumor expressed 50% or above PD-L1. So with that, I hope you got some idea of why we're testing coming from lung oncology at least. So the next question is, of these patients, who do we test? As I alluded earlier, we're trying to get clinical features that to select whom to test. So in the beginning, like dating back to earlier 2000s, we will find that the East Asian patient, female non-smoker, were concentrated with EGFR mutation. However, through extensive study in the West also, we find these patient group are all benefit from this testing. This include any lung cancer that has adeno components or those non-squamous. Therefore, except for the pure squamous cell type, we test them all. In addition, if you have this feature like they're light smoker or even non-smoker, or they're very young patients, we strongly recommend you test them. Recently, I received a very sad news. One of my colleagues is diagnosed with lung cancer. He's from China, of course, and he's East Asian. The first thoughts that pop up into my mind is we need a molecular testing. For sure, a week later, and he was found to have exon 19 deletion. So fortunately, we're able to start him on the osimertinib in 2 weeks of time. Move on to what we test. These are the list of the type of mutations we're testing, not exhaustive. However, the point mutations included such as EGFR or small insertions, deletion 19, and translocations, including ROS. The other features that may benefit from clinical decision-making include microsatellite instability, but these are less important other than colon cancer and the upper GI. Of course, there is strong evidence of supporting high tumor burden folks tend to have much obvious benefit when they receive immune therapy. Regarding the question of what to test in lung cancer, these are the mutations that have proven benefit that left out as the recent realization that RAF mutation is also recommended in the guideline. So with good evidence, if we find these mutations, the next level, we can use these different inhibitors in the management of our patient. These are the common mutations that you may find in the guideline in the near future, which I will not go through individually. The next, having find what to test, and then we want to know when to test. As Joe alluded earlier, and I think I summarized the timing into 3 categories. Of course, lung cancer as well as thyroid cancer has level 1 evidence of frontline testing. Therefore, before the initiation of first-line treatment, well, we want to test unless the patient is in a dire critical situation that does not allow us to have a 1 to 2 weeks of delay. The second situation we want to test is after the patient failed EGFR, especially the first generation or second generation EGFR inhibitors, because we want to look for the acquired resistance. So in lung cancer, those who received the first, second generation TKI has 60% chance of developing T790M, which we have a good drug that's already approved by the FDA. You all know that's osimertinib, so it makes sense to test. And of course, the third situation is when we exhaust all the clinical treatment options, we want to find out whether there's any particular somatic changes that we can target for clinical trial purposes. So as for how to test them, the first thing you want to think, you have to take these factors into consideration. You know, whether you have enough tissue and what's the tissue quality and what's the turnaround time, all these things. Do you have a bioinformatic analysis capacity? And after especially a large scale sequencing effort, how are we going to interpret it clinically to be meaningful? I can assure you the information you get from either the whole exome sequencing or whole genome sequencing is a daunting task. So these are the ways we do the test. It includes the pyrosequencing, which is the most frequently way of testing a single gene mutation like EGFR mutation there. They used the pyrosequencing method, which is pretty accurate. Apart from that, we can test for ALK translocation using even immune histochemistry. Of course, the best method to test for translocation for ALK or ROS is perhaps the FISH method, which enable you to see whether the two signals which are supposed to be apart come together to form a new gene or not. And if beyond these conventional testing now available are the next generation sequencing, we can use either blood or tissue to test. So if you have a tumor block, and then of course we can send tissue for analysis. And if for some reason rebiopsy is not an option and you don't have enough tissue left there, and you can choose the blood, the liquid biopsy, which Guardant 360 provides. That's also a way of testing. So, you know, because of the sheer, you know, profit that it's waiving, so you can see why so many companies trying to squeeze into this space. And I'm sure, you know, you're familiar with many of these, which I will not talk about. But the latest, I think this one is the Orion Project contributor there. But if you have a need to do the whole blood, the next generation sequencing, looking for resistant mutations, Guardant 360 is one to look into. So these are the currently approved lung cancer, you know, test with the Compania diagnostic here, and for which we have all different kind of small molecule inhibitors available, which I will not, I believe the majority of you are familiar with. So speaking of next generation technology, we have different level of testing. The commercially available test offers the spot assay. You know, during the test, they offer to test several hundred genes in which they have frequent mutations, and they have tools developed clinically for the management of patients. And they have, of course, the whole genome sequencing, during which you will have to test 3 billion base pair of a human genome, which is time consuming and costly also. Therefore, latest efforts is to test the whole exome, which is a collection of all the exons in the genome. Although it's also only about 1 to 2% of the whole genome. However, you will detect all the changes in the axon, which is responsible for about 80% of the somatic mutations that's causing human disease. So that's not too bad at all. For this kind of a test, the only thing you want to make sure is that there are enough a number of time that covered to make an accurate prediction that a reasonable chance that this alteration has a reasonable chance of being true positive. But there's also studies published using the low coverage to check the copy number variations to indicate which tumor will have a poorer prognosis, since the copy number changes in the peripheral blood is linked to the prognosis of that particular tumor. Next, I'm going to go into a little bit detail about the Foundation 1 test. Some of you might have sent out the Foundation 1 test and are familiar. But for those of you who are planning to do so, these are general requirements for the send out test. So they often require around 10 slides cut at 4 to 5 micron thickness, and then have an area of this to ensure at least 30% of, I would say, viable tumor areas. And in about 2 weeks of time, you will have hotspot sequencing about 300 to 400 genes coming back at a summarized format. They are covered about 500 times, and that in this test, they have reports about the point mutations, insertions, deletions, rearrangement, also about the tumor burden and microsatellite instability information. And they also provide complementary bioinformatic analysis and recommendation for FDA approved drug, plus suggestion for clinical trials. I'm sure you probably heard about the cost is about $6,000. Although this seemingly a high cost, however, if you find one that can provide the clinical benefit, actually compared to other conventional chemotherapy, it's worth it because you probably, those of us who are treating the patient clinically, even the conventional chemotherapy, the cost of one cycle of chemotherapy is much larger than this test. So I don't expect you to read this, but just to give you a taste of these the genes that's covered in the real testing, and these apologize for the poor quality of the slides that it's not written well, but I remind you that this is a summary from the report back, say what mutations they have identified through testing and what are not, and what are the interpretations. I'll skip this. If anybody who is ever interested in diving into the interpretation of the results, these are the clinical resources that you can use to look into, and that will help you to understand how they interpret it and the findings. However, as I said earlier, because of the sheer amount of information we get and the ever-changing guidelines, nowadays we need a molecular tumor board. So that's on top of the conventional tumor board, which we have the medical oncology, surgical oncology, radiation oncologist, and all these traditional players. On top of that, what's essential is this panel of experts who participate in the interpretation and recommendation of the further management, which include, of course, the molecular oncologist, pharmacist, bioinformatics expert, and even other experts, which I will not say individually. With this, you can think about, that we have quite a formidable task, and we have these hurdles to jump over for achieving this goal, and this including oftentimes small biopsies. In lung oncology, pulmonologists traditionally use bronchoscopy and fine needle aspiration, and a lot of times the diagnosis, the result turns to be non-diagnostic. Even if pathologists barely make the diagnosis, they don't have enough tumor that you can send out for this kind of testing. In thoracic oncology, in which we have a lot to test, we are often frustrated. And in addition, you have to figure out whether this was present in a germline or not, which oftentimes we don't have this information. In addition, we have the cost turnaround time issue, the access to clinical trial if you see patients in the community. Sometimes it's very challenging because lung cancer, especially after repeated chemotherapy, patient's performance decline quickly. Oftentimes, after several rounds of chemotherapy, patient is not in a situation they can withstand another biopsy anymore. So these are the challenges. So potential solutions, as I can think of, will include if we can have upfront comprehensive testing panels instead of sending out individual tests which consume a lot of slides, that will be something very beneficial for the management of a patient. And also, if possible, we always have in mind that we need ample tissues. So if you're using fine needle, make sure that at least you go multiple times. Of course, that's when you can ensure that the patient safety. Also keep in mind that this field is evolving quickly, and I myself is also involved in some of liquid biopsy. Therefore, stay tuned. In the near future, it may be possible to test for targeting mutations from peripheral blood. In addition to that, the other difficulty is to find with reasonable clinical evidence, when you find a mutation, when you give a certain drug to intervene, and they enough evidence to support the practice. Therefore, you all have a great role to play. You can help enroll the patient to make sure that we have good evidence coming out from clinical trial. Then we can use it in the real world. Shown here are the marvelous, very high specific and sensitive method my partner in Atlanta developed. So with this test, which is a quantitative time PCR method, he was able to detect from the peripheral blood with nearly 100% specificity and also 0.01% of sensitivity, which means if you put one copy of mutants in the background of 10,000 copy of wild type, that test will be able to pick up that. So right now, one project I'm engaged in is to sequence the tumor upfront when they have enough tumor to figure out which one could possibly appear in the peripheral blood, and design primers to go after the blood to try to find out whether that circulating tumor DNA is still there after intervention. Our hope is that by monitoring the change in the circulating tumor DNA level, we're able to figure out a better way to monitor the disease status, as well as design clinical trials for potential intervention. For example, if we detect the circulating tumor DNA level before lung cancer is resected, and then after the intervention, after tumor is gone, we're going to monitor that circulating tumor DNA. If this circulating tumor still remains, and then we can design the clinical trials, see if additional treatment like adjuvant chemotherapy will make a difference or not. So these are my collaborators from Atlanta. I always appreciated their collaboration. With that, I think I'm stuck here and take any questions you may have. Thank you for attention. Yeah.